future.
Alright, we're going live, guys.
Dude! That is so cool!
Weird Lowling vibes, right?
Oh my god.
And they drew me with my jacket.
Can they please make this into stickers?
Better, a shirt. We've already ordered them.
Oh, that's amazing. Wow. What a cool person that did that. You guys look great.
Evan, you're live on YouTube right now, okay? So
I don't know if I am. I'm trying to check it out on my phone and it's not popping up, and I've refreshed it. I refreshed the whole app.
Okay.
Oh, dude, that's because this is the new one. You haven't shared the, right?
I haven't shared the new one yet.
Okay, but we're live on the new one?
Yes.
Okay. Did you try to go back to the old one and see if...
10 people have already found us.
Oh.
Okay.
Oh,
You want to just say hello?
Hello.
Ask if people can hear you okay.
Oh, it's quite a delay.
Yeah, like a minute. It's not that bad.
Not that bad.
Considering 11 people are watching us at this point.
Yeah, if you like accidentally fart or something, we could hit cancel and you'll have like so much time. We'll be fine, right?
That was just.
No.
I think that's how it
I
works.
think that's just recorded.
Yeah, it is. That's why I'm saying we're live streaming. Just so when that happens, you can't be like, oh, shoot.
Oh. I'm gonna get away from the microphone.
I think the cat's a little freaked out by this. It's going. Where's that coming from?
What up? Here, see, I can take this down. Hold on. No, it's gonna be us soon. Hold on. Boom. Okay. So then wherever we put that, then they'll be able to. Oh, I got this all fucked up. That's hilarious. I'm not gonna move it because I have it all set up for the presentation. I don't want to fuck it all up, but this I could move where these red square is and bring that whole image into frame. I just have it set up this way because it's dialed in for that seat. Hey everybody, welcome to the chat. This is Portland Psychedelic Society. We're just had some technical difficulties trying to sign into our scheduled event. So we started this one. We're just going to be setting up here for about the next few minutes, and then I will check in with everybody. And if you could, share this link. We got Eisen in the house. He is preparing, and he's going to give his presentation on neuroscience. So just stick with us here for a few minutes. I'm gonna just put my head down and start sharing this link out. If you could share the link out as well, that would be awesome. Out to your psychedelic societies and your friends, and let them know this is happening. Thank you for finding this one.
Sweet. So, are we just gonna let it go until we're ready? Or we're gonna pause it?
Yeah, I don't think you can pause it.
Oh, cool.
So they're just gonna see us preparing, but same thing as Clinton Street, I guess. Like, you know, people see you moseying about and they're taking their seats. And
Yeah. Take your seats, everyone. Start cooking the popcorn now. So it's nice.
Oh, I have. Okay, here we go. I had the laptop mic off because it's going to be loud, whereas I got that microphone that he's going to be talking into that'll capture mostly just his voice.
Oh, nice.
So they probably didn't hear that announcement. Share the link. We're going live at 7. I mean, we're live, but we're going to have a real presentation at 7.
Jason, do you have any Scotch tape?
Yeah, maybe. Would it be okay if it were Irish tape? watching now, and we haven't even begun.
Hello, 19 people. What time time is it?
6:50.
We're almost there. Bye. Sorry, I missed you.
No, it's his cell phone.
No.
Oh, yeah.
Jeez.
How many cell phones have I bought? The years.
No wonder he treats them like that.
Yeah. This one I didn't.
Oh, that's good. You learned a lesson.
Yeah.
Right? I got a flip phone for him.
Me too. There it is.
Someone's ringing.
It's my ridiculous switch phone over.
I put a $10 or $15 budget on a Facebook ad that just says our in-person events are now online events. They are not canceled or postponed.
Sweet.
You know.
Right now. Is that connected to the our card?
No,
No, it's just
it pays me. That's yeah, right.
get reimbursed.
Yeah,
You think you will?
I'm allegedly supposed to.
I wonder how that works. Jason, can you fix it to where it will be for the thing? And can you put Aaron's presentation back so I can position accurately? That's where we're at.
Oh, I see. Terrence, a little machine elf.
How's that? Oh, there he is. Awesome. Maybe one of these two. Might as well.
Are we gonna play the jazz through the?
No, I'll turn it off. You should play it. I'll ask Aaron his preference. They probably really won't be able to hear it too much through his microphone, but when I cut my microphone on, then they'll be able to hear it. Or when I cut that one's really bad at picking up like the whole room.
Check, check. 1, 2, 1, 2. Microphone check.
Oh gosh. Yeah, it's fancy back.
And then then I can just hit you, boom, and then your mic is off. You look like completely in the shadows right now.
Yeah. I'll have to turn that one back. I mean, you could see me enough.
I mean, you just look like a figure.
Like a locked character. I can move to like.
It'll be fine because it's gonna start getting dark out.
Oh, okay.
It'll be fine.
So many messages.
How does that look? Well, let me check it out for myself.
Hey.
So what's going to be the signal for next slide?
Anything?
The what?
Signal for next slide.
Just, just the next slide.
Yeah.
And I'll try to just get a rhythm with you.
A dab.
We don't need to have, because you don't want to shout across the theater,
Yeah.
So, like, I'll just, okay. Okay, I'm going to cut my mic.
What we did next time was we were just like, next slide.
Yeah, that's fine.
I just feel like a little hand signal would be less. I mean, it's definitely not a big deal, but.
I feel like I can see everything okay on the screen here. Like when I'm looking on mine.
Can you see Terrence?
I can tell that he's there, but it's little.
Maybe move the Lysol.
Awesome.
No, we want them to know.
We want them to know.
But we're 5 minutes out.
5 minutes out. Whoop, whoop. Okay, so as far as the introduction, I have hi, welcome to the PPS YouTube live stream. My name is Evan Segura. I am the host of the Clinton Street Theater series, which is where this talk was supposed to be. But if you haven't noticed, the world is slowly ending. Too dark?
Too dark.
Not dark enough? Should I make it darker? Burning? Get your toilet paper, people. Seriously.
Okay, and then if you can get that as close to you as possible, it's real sensitive microphone, as in like, it's not picking up the whole room, it's only gonna pick up right up next to it. So the closer you be, the louder you'll be for the audience.
Do you think it would help if we took the...
You can take it off if you want, but it's like, you know, it isn't gonna give you too much more space.
Yeah, I'm fine like this. This is fine.
Yeah, as close as you can get it, you know.
Great. Okay, and then I was thinking about maybe talking about how we could really use some web design and program volunteers. If anyone has any web experience, fill out the volunteer form on the link tree, and the link tree is right there, so they can check it out.
I removed the volunteer form from the link tree today, and I will turn it right back on right now.
How dare you?
I know. I didn't think we were.
We're
I
getting.
didn't know if it was landing.
Yeah, I mean, there was definitely a peak and then a drop, but we just got to keep announcing it, and people keep filling it out. You know, we're building a study pool. Yeah, and then I'll just go through the PPS introduction, and then
Yeah, then I'll start.
I will finally be able to say you can solicit substances or services wherever you are watching this. We are no longer liable, and you do what you do whatever you. Oh, dang it! I do have to say it. Never mind. In person.
You are sitting in a room with a person who's also watching this right now.
You're sitting in a room of more than 10, spread out.
Give them there. Well, that's why I was like, it's gonna be funny because we'll be one at a time. Like, okay, 6 feet over here. Okay, there's Aaron.
Please say that.
6 feet. Hold on. Okay, hello.
And then 6 feet over here.
All right, it's 7, so I'm just gonna start. I'm just gonna do kind of the, I'm not gonna really overlap on you. I'm just gonna touch on that we're doing a live stream. I'm gonna do more of them, and also, that we're gonna do more online focused stuff, and that we need volunteers to help us manage Zooms and people with experience doing this kind of stuff. To just reach out to us. I made an email address, ppslivestreams@gmail, for people if they want to give us feedback or get in touch with us about this.
Cool. Sounds good.
So it's 7. I'm gonna hit the volume now and say hello. Hello, hello. It's 7 o'clock. Welcome. This is Portland Psychedelic Society's first ever direct to YouTube presentation. Everybody knows why we're quarantined at this point and doing things from the comfort of our own house and not at the theaters. But this is our way. We're gonna make sure that we, as we are a nonprofit dedicated to bringing information and educational events to the public. So we are going to continue on that mission by doing it online here. We're going to do all our support groups through Zoom meetings, and we're going to do plenty more here direct to online. So thank you for being here. I'm going to turn it over to Evan real soon. If you have experience, inspiration, ideas, feedback, or any of that stuff from what you see here tonight, we have an email address, ppslivestreams@gmail.com, and I'm going to turn it over to Evan and write that on the screen for you. And then we're going to get to Aaron Eisen real soon. Peace. hold on one sec. Okay, I got you on there. You're
Check,
so dark.
Microphone. Hi, everyone.
Okay, your mic is now on.
Okay. Hello. Welcome to the Portland Psychedelic Society live stream. My name is Evan Segura, and I am host of the Clinton Street Theater series, which is where this talk was supposed to be, but now it's in Jason's living room due to, obvious circumstances. Portland Psychedelic Society is a nonprofit organization dedicated to educating people about psychedelics, how to use them, how not to use them, where they come from. We are a 501c3 nonprofit, and like I said, we're an educational organization. We host events all over the city of Portland, except for right now, and probably not for a little while, but hopefully, once again, we will be able to. I was saying that this is the first time I haven't had to say no soliciting substances or services. Because you're not here right now, so you can technically do whatever the heck you want, but don't do it in the chat room. Let's keep that a productive space, let's keep things appropriate and hold that container for people. If you have questions, feel free to reach out through the chat room, and we're going to have intermissions throughout the presentation where Aaron will be able to answer some of the questions that you have. And so, a little bit about Aaron Eisen. He is the education chair of the Portland Psychedelic Society. He is a graduate student at the Natural University of Natural Medicine here in Portland, Oregon. He is also a neuroscience researcher at 2 labs at OHSU, correct? Yeah, cool. And he does a lot of great work here in Portland, including things like this presentation. So I'm very, very grateful to have you watching this, and I hope you enjoy.
Okay, I'm going to turn your mic on.
Peace.
Turn yours off. Cut to Aaron.
6 feet away.
Oh, I did miss that. Alright, let's give it a try. Say hello.
Well, hello everybody. Thank you for joining us tonight. I hope everybody's staying safe in these rather pressing times. But once again, everybody getting together to talk about this important message and kind of moving things through. So I appreciate everybody kind of coming here today. A quick background on myself. As Evan mentioned, I'm a graduate student at the National University of Natural Medicine. I'm also a research assistant at Oregon Health and Science University in the School of Nursing, Developmental Brain Imaging Lab, ADHD Study, the National Spina Bifida Registry, and the Portland Alcohol Research Center. I'm also a Resource Council member for Northwest Noggin, which is a K-12 public outreach organization. I am the director of the Translational Research and Psychedelic Studies nonprofit organization, TRIP. I'm also a student member of the Society for Neuroscience and an honorary member of the Portland State Neuroscience Club, but that's enough about me. Let's get to the slides. Next slide.
Next one. Oh, more?
Oh, no, that's good. So I'd be happy to send you these slides or answer any questions or even present research at your event free of cost. Generally, I would ask everybody to hold questions until the end of the lecture, but considering this is an online format, it would be perfectly fine if at any point you can put questions in, and we will be taking a few intermissions throughout the program. So at that time, I might be able to answer a few of your questions. And for any experts in the room, some level of generalization will be applied, as I won't be able to discuss absolutely everything about neuroscience and psychedelics in this hour and a half. Okay, disclaimer voice. The views and opinions expressed in this presentation are those of the author and do not necessarily reflect the official policy or position of OHSU, PSU, NUNM, Northwest Noggin, the Society for Neuroscience, the Imperial College of London, MAPS, and MAPS Canada. Next slide. So let's go down the rabbit hole. Next slide. So your brain is made of cells, and these cells are called neurons, and they carry electrical messages, and they connect chemically across synapses. And they send messages to each other with what's called a neurotransmitter. And there's this idea back when neuroscience was a evolving field, which it still is, but when it was kind of in its beginning stages, there was the idea of the sparkers and the supers. The sparkers believe that neurons only communicate via electricity, versus the supers thought that it was chemicals. And we now know it's both, and about 20% of your brain cells, the neurons, actually communicate strictly electrically. But it's kind of a combination of both, so they're both right and wrong at the same time. There was a very interesting question that was asked at one of my past lectures. It was like, okay, so these neurons send messages to each other. What do they say to each other? And they either tell the other neuron to turn on, which would be excitatory, or turn off, which would be inhibitory, and discontinue sending the message down the chain. Next slide. So on a neuron, there are these tiny proteins called receptors, and this seems to be the primary area where most substances seem to act. There's one specific receptor, a serotonin receptor, called the 5-HT, referring to serotonin, 2A receptor. And it seems that all psychedelics bind to this single receptor that is actually what defines it as a psychedelic. And the stronger it binds to this receptor, it seems the more powerful or potent the psychedelic is. And we can see this graph up in the top right of the screen. And we can see that to the left, it describes the potency. And on the horizontal axis, it describes how much a person needs. So how the affinity for the receptor, how well does it bind to it? And we can see that LSD has an extremely high affinity for binding to the 5-HT2A receptor, which suggests that it has a high potency. When I first saw I looked at DMT, and DMT was kind of all the way up there in the top corner, which made me kind of think that, well, that's odd. Most people think of DMT as being this holy grail of psychedelics, one of the most potent ones around. But if you think about dosage-wise, you might take 100 milligrams of DMT, but you would take maybe 100 micrograms of LSD. If you took 100 milligrams of LSD, it would be a much, much more intense experience than 100 milligrams of DMT.
Let's take a little pause here. There's a little bit of an echo. And they think that you're being picked up by another microphone, but I'm wondering if you're just not close enough to this microphone.
Okay.
Like maybe we can just pull this like right up to the edge here. I mean, you know,
Yeah,
it doesn't have to be that close, but that's pretty good. Try just saying a few words now. I know we're in a little bit of a delay, but
Testing. Does this sound any better? Testing.
1 sec, it's about to come through.
Oh.
It does still like sound like it's coming through like kind of like twice. It's not like feedback, but it's like a little bit of reverberance or something. What we can do is try just switching out. This microphone's way more just like picks up the whole room and doesn't need to be. Let's see. It's not going to be long enough, is it? Okay, give it a little test. 1 2 1 2.
Testing 1 2. Testing. Test.
Okay, we
All
should
right.
be good now.
We apologize for that delay, a few technical difficulties. Again, this is one of our first times doing a completely online, straight to live YouTube presentation. So let's get back to it. Next slide. So hallucinogens affect the gating of sensory input. There's a part of the brain called the thalamus, which is shown in the bottom left corner of the screen. We can see a human brain, and then we can see that small little island-like
object that's kind of in the middle of the brain. And that structure is called the thalamus. And what the thalamus does is messages come inwards through the thalamus and they go out to different regions of the brain. You could kind of think of it as the old telephone operator that would send signals or connect wires and connect two people together. So the thalamus does that. It takes information that comes from our brainstem and then sends them out to other regions of the cortex, as we can see that on the picture in the middle of the screen. This is courtesy of the Allen Institute for Brain Science, and we can see all the different projections that spawn out from the thalamus. Next slide. So psychedelics seem to shut down the thalamus. So when the thalamus gets shut down, that gating of sensory information gets distorted, and you kind of see all of these systems begin to increase in their connectivity, connecting to regions they might not normally be connected to. On the left half of the screen, we can see this circular figure, and we can see different colors in different areas of the figure. These different colors represent different brain lobes, and we can see under the normal brain, there's not too much connection between the different brain lobes. Yes, they are talking to each other, but not as much. But we can see under the influence of psilocybin, all of these connections start to flourish, and there's just this increase in global connectivity that occurs in the brain. This may be why things like synesthesia occur, the ability to kind of perceive, the ability to see sound or taste colors and other things like that, because different regions of the brain, sensory information, is now going to places it might not normally go to, like auditory information might go to the visual cortex, or so on and so forth. Next slide. Hallucinogens also affect a part of the brain called the locus coeruleus. The locus coeruleus is your orientation network. If I were to step out into the street and a car was coming toward me, my locus coeruleus would orient me towards the car, so I would have enough time to step out of the way and not to get injured. Same as if a spider ran across the floor, your locus coeruleus would orient you towards that to get away from it. So psychedelics hijack the locus coeruleus and make you oriented towards things you might not be normally oriented towards. Suddenly, regular stimuli becomes extraordinary. For example, the pattern on the rug suddenly becomes very interesting, or your hand, or that chair that's on the screen over there. Next slide. So where are these receptors? Well, they're all over the place. They're in the neocortex, which is kind of the outer layer of the brain associated with kind of higher cognitive functioning. It's in the olfactory cortex, which is very much responsible for smell. Hippocampus, which is linked to memory. The basal ganglia, which is loosely kind of generalized as being an important part of the reward network. The thalamus, as I explained before. The cerebellum, where interestingly a majority of your neurons in your brain are placed. We can kind of see on the brain, next to the brainstem, if we're looking at this picture on the screen, on the right half of the brainstem, we kind of see this very small object that looks like it's almost put in there with the cortex, and that is the cerebellum, and that is actually where about 69% of your neurons are in your brain. They're also found in the brainstem as well as the spinal cord, where information flows in. Next slide. Psychedelics also affect the default mode network, which is critical for self-reflection, self-awareness, and rumination. So psychedelics seem to shut down the default mode network, and this kind of promotes mindfulness and in-the-moment thinking. Your default mode network is your kind of daydreaming network. If you were kind of sitting thinking, oh, what am I going to do next week, Tuesday on my day off? Or what am I going to cook for dinner tonight? That is your default mode network acting. That might be why it's a little hard to plan what you're doing next Tuesday in the middle of an LSD trip. Interestingly enough, though, it seems that you don't need psychedelics to shut down the default mode network. Practices such as meditation seem to do the same thing, which may be why meditation is associated with mindfulness. Next slide. So first I want to kind of cover the different types of psychedelics.
It does not load on mine.
Oh, we had a few, I guess, more technical difficulties, but so there are a few classical psychedelics. If we could just maybe go to the next slide. Yeah, we'll just leave it there for a second.
Trigger warning.
Oh, there's the trigger. Okay. So there's different types of psychedelics. There's classical psychedelics. There's the empathogens, the entactogens, the dissociative, the delirians, and the research chemicals. So I'm going to kind of go over how these substances impact the brain in different ways, and I will be showing pictures of some of these substances, so if this would kind of bother you, this might be a good time to step out. Then again, it might not have been the best idea to watch a Portland Psychedelic Society presentation if you can't see substances. Next slide. So there's the classical psychedelics. There's LSD, DMT, psilocybin, mescaline, and ibogaine. And these are all 5-HT2A agonists. And when we start in the top left of the screen, we can obviously see that's mushrooms. The next one over would be DMT crystals. Following that would be pure ibogaine extracted. Following that would be pure mescaline extracted. And below that would be blotter art. Next slide. So then there's the empactogens and entactogens. So there's MDMA, MDA, MDE, pretty much all the alphabet soups that come together. So these act as reuptake inhibitors for serotonin, which is exactly the same mechanism that SSRIs tend to do. So basically, serotonin is released into what's called the synaptic cleft, which is a gap between two neurons, and these are where the receptors are. And then once the neurotransmitters enter this gap and go in there, they bind to receptors, and then they get sucked back into the neuron for which they had been released from, and they're either repackaged or destroyed. So SSRIs block the reuptake, so the ability for the neuron that sent the serotonin to suck it back in, which creates more serotonin in the synaptic cleft binding to more receptors. So essentially, MDMA and MDA and the other empactogens and entactogens act as SSRIs. Furthermore, they also they act as reuptake inhibitors for norepinephrine, which is actually the same mechanism as SNRIs, which block both serotonin and norepinephrine. And they're also weakly reuptake inhibitors for dopamine as well, which actually has the same mechanism as cocaine, which is kind of interesting. They're also known to promote an oxytocin release, which may be why we kind of see these positive mood, kind of openness, and willing to kind of go and express and communicate more with people. Next slide. So then there's the dissociatives, such as PCP, ketamine, DXM, which is common cough medicine, and salvia. Now these are all NMDA receptor antagonists, so they actually block the NMDA receptor. And the NMDA receptor is thought to be a very critical component of learning and memory. There was a very interesting experiment where they took a mouse, and basically from past research, they knew that these NMDA receptors are open for longer periods of time in younger animals and humans. And they're open for longer periods of time, so it helps a person to learn when they're younger. And then as a person grows, these receptors become less reactive to stimuli. And again, in that experiment with the mouse, they took a young mouse's receptors and they put it in an old mouse, and the mouse was much better able to navigate through a maze. They also affect the opioid receptor, so they're agonists for opioid receptors, which may be why PCP, ketamine are kind of used as anesthetics. Interestingly enough, PCP was initially developed as an anesthetic for surgery, but there was definitely some issues around using it for that. And ketamine is currently commonly prescribed for animals because they can't complain about having negative effects from it. And it's interestingly enough, it's also prescribed quite commonly for women who are giving childbirth, because that's a time where you generally don't want to feel like you're in your body. And dissociatives affect the brain called the parietal lobe, which if I were to close my eyes right now, I'm able to touch my nose. And this is through my parietal lobe. It's kind of the mapping of where I am in the world. And this seems to be interrupted under the influence of dissociatives. There was a very interesting experiment where they took somebody who was awake and they operated on their brain, because you don't have pain receptors in your brain, so somebody could be conscious with their brain exposed. And they used a small electrical stimulation to a specific network in the parietal lobe, and the person described feeling like they were being lifted out of their body. And then when they took the electrical current away, they got sucked right back in. So dissociatives seem to affect this particular network in the parietal lobe. Next slide. There's also the Delirians, which are nutmeg, Benadryl, Dramamine, deadly nightshade, and datura. These are all anticholinergic and antihistamine. Most of these, if not all, are legal, although it's kind of concerning. Most of the Delirians are some of the most dangerous psychedelics. Acetylcholine, which refers to anticholinergic. Acetylcholine is the neurotransmitter that seems to be found most commonly at the neuromuscular junction, where muscles connect to the neurons to send messages and other factors like that. So too much of any of these could essentially disrupt your heart and lungs and breathing and a whole bunch of other factors, and they could be quite deadly at higher doses. Next slide. And then there's the research chemicals, which are, I call them pseudo-psychedelics, but definitely needs to be more attention kind of put towards these. These are very dangerous substances. We do not know what the long-term effects of these substances are. Some of them have been developed in the 1980s, but some of them as recently as 2013 and even newer than that. These chemicals are not meant for human consumption. Most of them, like such as 2,5-I, was never designed to be consumed by humans. It was designed to map the 5-HT2A receptor. And because of this, they can be very dangerous to consume because we don't know the long-term effects. People have been taking LSD for 60, 70, 80 years now. So we know the long-term effects from LSD and cannabis and mushrooms, but we do not for research chemicals. Interestingly enough, on one of the pictures, most research chemicals are, again, another danger about that is that they can be sold as psychedelics. So that really stresses the importance of testing kits to make sure you know what you're actually consuming. They can be very cheap to purchase online, and the picture that's kind of in the middle, no worries. The picture that's kind of in the middle, we can see these kind of blotter tabs, and it says 2,5-I and quotation marks plant food. So some vendor online is selling these chemicals as plant food. I don't know what kind of plants they're growing, but that is definitely something to kind of be wary of when you're consuming psychedelics. Next slide. So if you want to learn more information about psychedelics, I highly recommend checking out Erowid. They're a substance encyclopedia that has lots of very useful information about a whole bunch of different hosts of psychedelics. Next slide. So the 5-HT2A receptor is the main excitatory receptor subtype among the GPCRs for serotonin. And what GPCRs are is there's two main types of receptors. There's called ionotropic, which opens when a drug binds to it, they open, and chemicals go inside, ions such as sodium, potassium, and chlorine. But then GPCRs, they're a receptor with a small piece on the inside. When a drug binds to it, they click off, and they go inside the cell and send other messages inside. And the 5-HT2A is generally excitatory, so meaning it sends messages down to other neurons. It keeps the messages going. Although interestingly enough, the 5-HT2A may also have an inhibitory effect on the visual cortex, which may actually explain why we have hallucinations from psychedelics. Whenever there's a lack of information, our brain reaches out to other regions to pull that information in. That is generally why there's the occurrence of things like phantom limbs, where a person who has lost an arm or a limb experiences a hallucination that they still have that arm. It turns out that regions of the brain that were responsible for picking up signals from that arm actually branch out to other adjacent areas and pull information from that. So whenever the person moves those adjacent areas, it registers as the arm. There's a similar effect for psychedelics in producing hallucinations.
Hold on, before you start talking, just a little bit lower, just say, hey, we just raised the audio on the mic, so you might have to adjust here.
So we just raised the audio quite a bit on the mic, so might need to adjust a little bit.
People mentioned the lighting. You look a little spooky.
Okay. All right, yeah, changes as the sun's going down here.
Yeah.
Okay, this is a learning curve.
It is.
You're getting a little shadowy. Yours was you were just a complete shadow being.
Darker now. Yep. I think we should just turn this one on, dude.
Turn that one on.
Yeah.
Okay.
Hold on for a few more technical difficulties.
Here we go.
Technical exploration.
You look great. You look great.
Okay.
We're learning.
We appreciate you working through us with these technical
How does it
difficulties.
look on there?
It's good.
You look great.
Much better.
Okay, we're all good. Let's please continue.
All right, next
grab the
slide.
Right slide.
Yeah.
Sweet. Oh, this one too.
So we seem to be having some issues with the formatting of the slides, but I'd be happy just to read them off to you. So the 5-HT2A is expressed in the high levels of the cortex, which is responsible for learning and memory retrieval. It's heavily concentrated in the postingular cingulate cortex, which is a central mode of the default mode network, which may explain why psychedelics have such a substantial impact on the default mode network. They are very scarce in the primary motor cortex, which may explain why they have such a strong mental effect, but barely affect movement. And it seems that this receptor is not just there for psychedelics, surprisingly as that is. It has a natural purpose, and its purpose is when we're in times of stress, the system activates, and this promotes enhanced cognitive flexibility, improved dissociative learning, and hippocampal neurogenesis to help us to be able to adapt to situations. Next slide. So for the subjective effects, we have well-being, optimism, openness, ego dissolution, and it's very helpful when there's a need for flexibility to break rigid thought patterns.
It's like shooting me. Okay. Personality.
Yeah. So there was a recent study done that looked at psychedelics and spirituality, and they found that psychedelic use was associated with more liberalism and eco-friendliness. The study found that specifically psychedelics were associated with increased openness, and this was not found with cocaine and alcohol. Psychedelics also were found to increase nature-relatedness, and alcohol actually decreased nature-relatedness, when nature-relatedness was defined as, I always think about how my actions affect the environment. And when I think of liberal and eco-friendly, I generally think of Portland, which I'm not saying psychedelics help to make Portland that way, but it could be why Portland is such an attractive hub to people interested in psychedelics. There was also a study that came out in 2014, and they looked at psychedelic use in criminals. And they looked at recidivism, which is the probability that, or when somebody is released from jail, they go back to jail. And they found that psychedelic use was a greater predictor against people going back to jail than stable family, housing, and even employment. Next slide. So there's been a recent trend and interest around microdosing. So I want to kind of discuss the difference between high doses and low doses. So first I want to define two terms. There's kind of convergent thinking, which is persistence over flexibility. And then there's more divergent thinking, which is flexibility over persistence. And in a high dose, we see an increase in divergent thinking, or kind of this flexibility, a more liberal or open mindset, and a decrease in convergent thinking, which is more persistence over flexibility or a more conservative mindset, but also an increase in creativity. However, on a low dose, we see an increase in both divergent thinking and convergent thinking creativity, which is very interesting. Next slide. So it seems that microdosing helps our mind go towards the most efficient zone, and we see enhanced motivation and focus, and reduced distractibility, procrastination, and rigid thought patterns or behaviors. It seems that the 5-HT2A receptor has an indirect modulation of dopamine, and I see the graph might be a little distorted on your screen, but it kind of has an inverted U-shaped graph. So where there's very low dopamine levels, performance is low. But when dopamine levels kind of reach the middle range, so not too low or too high, kind of the Goldilocks factor, you reach this kind of optimal level of dopamine, which helps to promote cognitive thinking, which may be why we see people in the Silicon Valley and people who have depression treatments using microdoses. Then when somebody kind of takes more, they kind of have this reduction in performance and function, but we can kind of think of that as almost a reset. And there might be a good application for both microdoses and high doses. Maybe with high doses, it would be more effective in the treatment of things like addiction. Next slide. So I recently went to a talk with Dr. Robin Carhart-Harris, who is the head of the Center for Psychedelic Research in the Division of Brain Science at the Imperial College London. He's currently leading the field right now in psychedelic research, and I'd like to discuss some of the things he shared with us at that lecture. Next slide. So serotonin is involved in neuronal plasticity, and neuronal plasticity is the brain's ability to adapt or change to its environment. Next slide. It seems that serotonin is heavily involved in the sensitivity to context, environmental factors, and research in humans and animals has shown that manipulating serotonin modulates an organism's sensitivity to the environment. As we can see in the graph in the bottom left, if there was no plasticity, organisms' traits would be independent of their environment. But we can see when there is plasticity, an organism's traits begin to change based on the environment they are in. And this helps to promote the most adaptable and efficient form for that organism in the environment. Next slide. So when we're talking about serotonin in the environment, we have to realize that there's different types of environment. There's both enriched environments and there's adverse environments. And we've seen that increasing serotonin in more enriched environments increases the positive effects of the enriched environment. However, increasing serotonin in a more adverse environment seems to increase the negative response to that environment. And when we think about SSRIs and how they work, their primary goal is to increase serotonin. However, maybe this is why SSRIs don't work for everybody, because it's really dependent on the environment. And even though SSRIs have shown to be very effective, we got to consider who has access to treatment, possibly people in more enriched environments. And when we're thinking of enriched and adverse environments, it's a little more complicated when we work with humans, but when we're talking about mice, you could think of a more adverse environment kind of being like the small empty bear cage on the screen, versus a more enriched environment is kind of to the right of the screen, where we see lots of little wheels and little toys and all kinds of different things for the mouse to do. There was a very interesting experiment called Rat Park, in which the researchers took mice and they put them in the bear cages and they gave them a choice of morphine, water with morphine in it, a morphine solution, or just regular water. And the mice would drink the morphine solution until they died. Then the researchers had an idea. They're like, what if we created a rat Disneyland? Make it very open, no cages, lots of room to run around, lots of friends to play with, a whole bunch of other like cheeses, a good old time. And they gave the mice the same choice, the morphine water or the regular water. And when the mice were in the more enriched environment, they barely touched the morphine water, which is another very interesting result. Next slide. So serotonin is involved with mood. It's a complex relationship that depends on the context of an environment. Increasing serotonin does not necessarily increase positive mood, which really stresses the importance of context. Next slide. So what happens to the 5-HT2A receptor under psychedelic use when it's stimulated? We see an increase in low-level associative learning, an increase in extinction learning or unlearning, and an increase of flexibility and thought patterns. Physiologically, we also see an increase in neuroplasticity in the cortex, so kind of the growth of new neurons. And we also see synaptogenesis, which is increase in functional components of neurons, which would be referring to the axons or the dendrites or the axon terminals, kind of the regions that branch out and connect to other neurons. Next slide. So the 5-HT2A receptor is expressed highest during younger periods of brain development. And this allows for more plasticity early on when an individual is learning so much, such as a sponge sucking up and absorbing new experiences. We're actually born with about 200 billion neurons, but by the time we are 24 to 26, we only have 86 billion neurons left. We lose almost half of our brain cells through development. Whenever we go on outreach with kids, we always like to joke around that school kills your brain cells. But it turns out those brain cells die off because you don't need those networks. You're born with more networks than you need. And depending on what experiences you do when you're younger, your brain says these networks are more important, keeps the networks you're using more, or it will remove the networks you think are less important. So the 5-HT2A receptor actually plays a very key role in allowing us to observe new information to kind of help with what's called synaptic clipping. Next slide. So under the influence of psychedelics, we kind of see this breakdown of specialization as the brain gets older and you kind of lose the unnecessary networks. The networks that remain become more segregated and increase in their ability to function. This is why an infant can barely do anything versus an adult is much more functional. However, under the influence of psychedelics, we see a system regression. Developmentally older brains actually start looking like younger brains. We see the global integration, as we saw earlier on the slide that describes psilocybin in the brain. We kind of see all these networks start connecting. And we also see a local disintegration. So these networks that were highly specialized in doing one thing start to break off and help support other networks. And the brain kind of functions more as a whole. Next slide. So again, as I mentioned before, the 5-HT2A receptor is not simply there for psychedelics. It has another reason. It's there to respond to stress, and this allows us to be more adaptable when things are stressful. This is especially when we need to be thinking on our toes in order to survive. This is when the system engages, and it's useful and improves our chances of survival. Next slide. So now we're going to begin discussing psychopathologies and psychedelics. This may be a good time for a brief intermission. So if you got to get some water, want to get up and stretch, we're just going to do that. And then we'll come back in about...
just a few minutes, and we'd have maybe opened a question or two from the audience. There really hasn't been any, so...
yeah, we'll maybe come back in 5 minutes. And if you have any questions, I'll go over a few of them before we get started on this next section.
That's, we're in the shot, but if you get up where none of us are on camera, and they shouldn't be able to hear us. But we'll have to live monitor that situation. Yeah, I've been watching the chat. The only question that came up, one guy asked where he could buy mushrooms in Portland. Jesus, we just said it.
Actually, I thought I saw another one.
Yeah, they can hear us.
They can hear us now?
Yeah.
Oh.
I don't know how. I don't know what mic it's coming.
A question from Jesse F. Do you think that you experience everything during the pain, but just lack the ability to remember? I don't know what that was referring to. It was at 7:21 p.m. So maybe we could see.
Yeah. Where was it? 7:21. See, it's still, the audio's still on from somewhere.
Oh boy.
So that's what they were talking about.
Stop listening to us.
Watching us.
Yes.
I don't know where the audio's coming from.
Also, there was a gentleman, Mark, that thanked for the growing class on YouTube. Just want to say you're welcome, Mark. Glad you enjoyed the video.
That's all. Here, do you want to be on there?
Oh yeah, sure. I got a few things to say. But I forgot to say in the intro. Oh boy, someone's calling me. Casey just called me. Could you get in touch with her? Yeah. Hi everyone. So yeah, a few things that I wanted to say.
Dang it, now I can't remember. I remember one of them. One of them was, if you go to the link tree that you could see on the screen right now. Did you put the volunteer form back up?
I'm gonna do it right now.
Okay. Either way, you can click the website. You can click the website, and the website, you'll be able to see that there is a volunteer form on there. If you have any experience with web design or programming or anything like that, please fill out the form and help us, because as you can see, we're definitely struggling a little bit with the transition from hosting our events to going online. Nothing I don't think we can handle, but any help would be greatly appreciated. This is a really strange time to be alive during all this craziness with coronavirus. And we don't know how long we're going to be online, but we do know that it is essential that we continue to nurture this community here in Portland and globally. That being said, if you're watching this and you're not in Portland, Oregon, and you're in a town or a city that doesn't have a psychedelic society, I highly recommend that you consider starting one. It is seriously as easy as making an account on Meetup, finding a cool coffee shop or meeting place, and then meeting up there maybe once or twice a month and just getting people together. It is so important to provide this container for people to come together and talk about their experiences, good or bad, and integrate them with a supportive community that can help. Jason and I have been talking a lot about this new idea of kind of creating a guide to start your own psychedelic society, and now that we're not hosting any in-person events, we have a lot more time. On that note as well, we're going to be recording a much more accurate and thoughtful video on how to grow mushrooms, because the ones we have on YouTube right now are informative, but it's a open Q&A discussion, which I have seen is a bit frustrating.
I said that.
That's about all I got. If you feel moved to volunteer, especially if you have web experience, please get in touch with us. And if you're not in Portland, Oregon, please consider starting something like this wherever you are. Like I said, weird time to be alive. Not only because of coronavirus, but also because you're watching someone teach you about how psychedelics affect your brain on YouTube with like 100 other people right now, which is really strange. And it's also really cool. So thanks, everyone. Hope you're enjoying this.
Okay, volunteer form is back on.
Whoop, whoop. Okay, I didn't really see
If there's any questions, guys, now's the time.
Now's the time to put your questions up if you have any about what Aaron's covered so far. If you have other questions, save them, because he might cover it in the next 30 minutes. So
there's a 1 minute delay or so too, so lighting looks good.
I think we're good. I'm gonna pass this.
All right, I'm gonna get a cup of water. Yeah, if you just want to get yourself reestablished here.
Sure. Just let me know when to start.
Please stop asking for where to get psychedelics.
Okay. If you ask how to get these, like, the answer is you have to figure that out on your own. And if you ask, you're gonna get banned from the channel.
Is there a Zoom option?
Yeah, it's this. But you gotta pay attention to this. Oh, okay, there you go. This is the monitor.
Oh.
You actually had it perfect,
Oh.
however you had it. But, okay.
I'll give it back there.
All right, Aaron, go ahead.
All right, let's continue on.
I'm going to get you a little water, so you might hear that. But
Okay, next slide. So we're going to be covering current research that's out on psychedelics, specifically MDMA to treat PTSD and psilocybin to treat depression. There's been a lot of research right now around both of these. We have Compass doing phase 3 trials for depression with psilocybin, and MDMA with MAPS has currently just started their phase 3 trials as well. Both MDMA and psilocybin have been given breakthrough therapy by the FDA for treating PTSD and depression. In 2017, it was MDMA. In 2018, it was psilocybin. Next slide. So first, MDMA to treat PTSD. Next slide. So this lecture was done by Mark Hayden, who's the director of MAPS Canada. He's an adjunct professor at the University of British Columbia, and he has 28 years of experience in the addictions field. He also has publications on issues of drug policy. Next slide. So classical psychedelics offer a sense of generic spirituality. And what I mean by generic spirituality is Christians find Christs, Buddhists find the Buddha, and atheists find the entire universe. Whatever your belief system is, that is what is manifested. If you believe in the vine, you will find the snake in the vine. Next slide. And this is incredibly helpful in the treatment of many issues. There's a disorientation of ego when fundamentally one's self of sense changes. And this is very useful for the treatment of alcoholism. If it's one's ego that is persuading one that it is acceptable to drink 26 ounces of vodka every night of their life, that belief system needs to be disoriented. Next slide. And classical psychedelics are useful for this. They are unconscious amplifiers. They help us to access materials in our unconscious mind. The permeability between the conscious and the unconscious mind becomes less. We can dig deeply into ourselves and find stuff. And if issues are buried deep within our unconscious minds, we can find them and we can rework them. Next slide. There's also the portal effect, the wow, that was incredible effect. Like climbing Mount Everest or graduating from a university, it's a major sense of accomplishment, achievement, and transition. And that is useful for treating substance use disorders and helpful with withdrawals and detox, as well as depression and anxiety disorders. Next slide. Then there's the empathenogens, which promote connections with others. Those include MDMA, MDA, and 3-MCC. And these kind of promote an intense bonding. And then you have entactogens, which are more the connections with oneself. Next slide. There's also the therapeutic alliance. And the therapeutic alliance is the greatest predictor of success in any type of psychotherapy. And these are alliance-building medications. They help one to reflect on oneself. They reduce the fear of PTSD treatments. PTSD is like a tape loop that is buried in our unconscious minds. As traditional therapies get close to the tape loop, there's a huge fear response, and MDMA reduces that fear and allows people to confront that tape loop. As such, MDMA is a state-of-the-art treatment for PTSD. Next slide. So Michael Mithofer, MD, wondered, how is MDMA-assisted psychotherapy different from other types of psychotherapy? Next slide. So he went to the Canadian Military Health Conference in Vancouver, British Columbia in 2018. And at this conference, scientists were pitching PTSD treatments to the military. They claimed at best a 25% level of effectiveness. However, the literature says closer to 10%. It takes years and the dropout rates are high because people don't want to re-experience their trauma through flooding or prolonged exposure. Sending a war vet to re-experience their trauma and desensitize to it is very difficult. It's a challenging process. Next slide. So Michael Mithofer demonstrated an 83% level of effectiveness for MDMA-assisted psychotherapy, from 10 to 25% to 83%. Now, if you're a researcher and you see a jump that big, you have several thoughts. They're lying. They're falsifying their data. And generally in this field, normal progress is at best in 5% increments. So MAPS took this idea and they reran the study in a larger trial with a greater group of therapists with a more diverse population under the supervision of the FDA, and they demonstrated a 67% level of effectiveness. Next slide. Here is the paper that was published on this. If you are interested in me sending you this, please grab my email or I'll probably restate my email at the end of this. So if you have any questions that come up afterwards or you want access to these research papers, feel free to just send me an email and I'll send you these papers. Next slide. So now let's cover psilocybin to treat depression. Next slide. Depression is a leading cause of disability among American adults. It's currently the third leading cause of disability in the United States. 16 million Americans had a depressive episode in 2015, but I wonder how they got that statistic. You can't survey absolutely everyone in the United States. So I believe that is kind of a rather low estimation. Next slide. So Robin Carhart Harris set out to do a study to assess the safety, the feasibility, and the effectiveness of psilocybin in treating depression. So in this study, there were 20 subjects who had what's called treatment-resistant depression. I don't like that term though, treatment-resistant. It sounds like they're resisting against the treatment. Well, no, the treatment didn't work for them. It's not that they're resistant, it's that these medications did not help. And they define treatment-resistant depression as a failure of 2 classes of medication. However, in this sample, the average age of depression was 20 years. So these participants had been depressed for a majority of their life. And it's likely they had tried more than just 2 classes of medication. They'd probably tried every available psychotherapy, medication, anything that they could get their hands on in those 20 years. So they used the Quick Inventory of Depressive Symptomology, the QIDS, to assess depressive symptoms. And this was a 1-week experiment with 2 psilocybin sessions. First, there was a small dose on day 1 of the study, and a large dose on day 7 of the study. Next slide. So here's kind of a quick map I drew out of the study. It's not the exact dates the study took place, but it's just to get a better idea. So on day 1 of the study, which would be June 2nd on this calendar, they were given a very small dose of psilocybin. And this very small dose was not meant to have any necessarily therapeutic effects, but just to see if they could tolerate it, if they didn't have any adverse reactions, just to see kind of a safety dose, if you will. Then they didn't give them any psilocybin for the next 6 days. And on day 7 of the study, they had a high dose, which was the therapeutic dose. And they did a brain scan immediately following the high dose. 1 week after the study, every participant experienced reductions in their depression. And 3 weeks after the study, 9 of the 20 participants were in full remission. So they no longer had measurable levels of depressive symptoms from a single therapeutic dose of psilocybin. Next slide. So now that when they looked at the brain scan, I see the slide's a little distorted, but we can still work through it. So when they looked at the brain scan, they looked at a region called the amygdala. And the amygdala is it's a part of the brain that's looked at a lot in psychology. It has a pretty big association with emotional responses, specifically fear responses. It seems to play a big role in. Like if a spider was running across the floor, your amygdala would make you, I want to get away from that. So amygdala seems to be, as I mentioned earlier, less reactive under the influence of psilocybin. So when somebody takes psilocybin, their amygdala starts responding much less. But interestingly, 1 day after a person consumes psilocybin, the amygdala increases in reactivity, which is very interesting. And that's the polar opposite goal of SSRIs. SSRIs tend to try and increase serotonin in order to decrease amygdala's reactivity. And a common psychological test used to assess depression when using brain scans is fear face response. So we can see in the bottom right corner of the screen, there's a woman smiling and then a woman having a more fearful face. And participants go into a brain scanner and they look at these faces, and then researchers look at their amygdala's responses to the faces. And people who have greater levels of depression generally respond quite similar to both the happy and the negative faces. So they found that SSRIs increase the response to both fear faces and happy faces, which is the exact opposite of SSRIs. And the researchers kind of quantified this as the acceptance of all emotions, both good and bad. It's important to consider that these participants had been depressed a majority of their life, about 20 years. They were very familiar with their depression. Maybe their kind of negative emotions increased slightly, but so did their positive ones too. They were once again able to feel positive emotions, and that might have made all the difference. Next slide. As mentioned before, psychedelics also have a pretty substantial effect on the default mode network. When somebody is consuming psychedelics, the default mode network tends to get shut down. However, one day after psilocybin, the default mode network increased in functional connectivity. So again, we saw a polar opposite effect for when somebody consumed psilocybin to afterwards. And then 5 weeks post-treatment, they did another brain scan and found that there was actual spatial increases in the default mode network, but not for every participant. Only 9 of the 20 participants had spatial increases in the default mode network, specifically those 9 people who were in full remission. So it seems like the spatial increases in the default mode network is very much tied to the clinical outcome, to reduction in depressive symptoms. And increase in spatial connectivity of the default mode network is a similar therapeutic goal of electroconvulsive therapy. But if you'd ask me, I'd rather eat a mushroom than go through electroconvulsive therapy. That might just be me, though. Next slide. So the subjective effects of mushrooms, they increase cognitive flexibility, and they increase well-being and optimism. There's also an increase of mystical experiences, which also seems to be associated with increased psychological well-being and a decrease in depressive symptoms. Next slide. So one of the greatest predictors of success in the treatment of psilocybin is the amount that people have a mystical experience. The degree of mystical experience is often highly correlated with the success of the outcome. People with the most mystical experiences have the greatest positive treatment effects. But how do you measure mysticism? Well, there's the 30-item Revised Mystical Experience Questionnaire, the MEQ30, which was validated by Barrett, Johnson, and Griffiths in 2015. It's currently one of the most commonly used mystical questionnaires to date in research. Next slide. So there was one study that looked at mysticism in participants who consumed psilocybin. Participants were given a high dose of psilocybin, or surprisingly, methylphenidate or Ritalin control. And this is very interesting because most of the time it's very hard for researchers to use what's called a randomized controlled trial, which is the gold standard of research in psychedelic studies. Because it's very hard to have a control group in psychedelics that doesn't take psilocybin or a psychedelic. Most people would be aware if they were given a placebo and they weren't having psychedelic effects. So they overcame this by enrolling people who had never tried any psychedelics before, so they didn't know what they should feel like, and they gave them what's called an active control. So they didn't just give them a sugar pill, they gave them a medication that had effects. So it would be harder for the participants to know if they were in the treatment or control group. They did a pre-post blind test. So they measured both before and after, and participants did not know which group they were in. They measured the participants' psychological well-being and mysticism 2 months and 14 months after a single psilocybin dose. And they asked the participants about positive mood changes, an increased sense of spirituality, and how personally meaningful the spiritual experience was for them. Next slide. So this is the study. Again, if you take my email down, I'd be more than happy to send it to you. Next slide. So what they found was the numbers went up. So before they consumed psilocybin, they had a rather low level of mysticism. This increased 2 months later, and it increased even more 14 months after a single dose. Next slide. And this is a groundbreaking study with amazing results. Can you think of any intervention a psychologist, psychiatrist, or doctor does to anyone that you wait for up to 14 months and remeasure, and the number goes up from only a single treatment session? The researchers found when administered under supportive conditions, psilocybin occasioned mystical experiences were considered by volunteers to be among the most personally meaningful and spiritually significant events of their lives. Next slide. So why did this treatment work for you? When they asked the participants that, the participants responded that they went from disconnect to connect, the avoidance of difficult emotions to the acceptance. And they felt that CBT and antidepressant treatments kind of reinforced this disconnect. They felt like they were being handed from one therapist to another, from one medication to another, without any real benefits. And this can be very discouraging to people who are dealing with these issues. Next slide. So a couple metaphors that come to mind to describe a psychedelic experience are the therapeutic benefits that occur after the psychedelic experience, kind of a calm after a storm, a broader perspective, a reset. Next slide. Kind of flexibility, mind lubricant, if you will, and reconnection. Next slide. And I like to think of it kind of the acute state of psychedelics. When somebody is consuming a psychedelic, it's like hot molten metal. That is generally not the time therapy is applied. But it seems after psychedelics, the effects wear off in the following days, weeks, and months. And when we kind of see these neuronal changes inside of our brain, that it seems to be almost the best time to apply therapy. When a person is kind of like a more liquid metal and they're able to be molded and adapted to different situations to increase the effectiveness of their therapy. Next slide. So now I want to talk a little bit about the dangers of psychedelics. This actually might be a good time to take another short intermission.
Cool, we have a bunch of questions too, so.
Awesome.
I don't know any way of killing our audio, but Evan, if you want to get back on, or here's the questions.
Sure.
We can field them in any order.
Sure.
One question, difference between an entheogen and a psychedelic. Okay. Another one asked about what is negative affective bias. I think maybe a term you used. Okay. And then another one was, that's going a little discussion right now, can you microdose with MDMA?
Okay.
So I don't know if you want to take those now.
Yeah, sure.
Or...
I'll answer a few of those now.
Let him get this situated. How's that? Pretty good.
So all of the chemicals that I discussed here are psychedelics. It's just, and pathogens and adaptogens such as MDMA and MDMA are a type of psychedelic. Generally, we think of a psychedelic as being one of the classical psychedelics, which is like DMT, ibogaine, kind of other things like that. But MDMA is still considered to be a psychedelic. It's just in a specific category of psychedelics. Such as like PCP is actually considered to be a psychedelic as well, although it's also considered to be a dissociative. Substances can have more than one type to it. For example, cannabis is considered to be a stimulant, a depressant, a euphoric, and a psychedelic all rolled into one. So substances can kind of have different effects to that. But yes, again, to answer the question, impactogens and entactogens are all types of psychedelics. They're specific types.
What about, the question was entheogens.
Oh, entheogens. I'm sorry, I misheard that. So entheogens are definitely psychedelics as well. They're just referring to the psychedelics that are more used for kind of ritual purposes or kind of less recreational, like I mean, even MDMA is not necessarily used for complete recreational, but generally when you think of the entheogens, you think of like ayahuasca and kind of ibogaine and other kind of factors like that. They are definitely psychedelics as well. They just kind of, they don't really fall into a specific class, more the classical psychedelics, if they would kind of fit into there.
Okay, and then the other discussion about microdosing MDMA.
Oh, absolutely. So microdosing MDMA definitely has been explored. Actually, when MAPS was doing its trials, they did a placebo control trial. So basically, they had participants who were given the full dose of MDMA, then they had a control group. And the control group was actually also given MDMA, just a very smaller dose, or a microdose, if you will. And the results of their studies showed that the higher dose of MDMA was much more effective than the lower dose. And kind of on lower doses, MDMA might not have any substantial effects. It might more have kind of these stimulating effects and kind of elements like that. It might be beneficial for some people, but the research is really showing that kind of these higher doses are where kind of the therapeutic relevance is when we're speaking in terms of MDMA.
Okay, and then thank you for that.
That's the right one. Is that
Yeah.
Yeah. Okay. Cool.
The guy wants to hear about the neuroscience from a neuroscience perspective. Ask him how SSRIs interact with psychedelics.
That's a very good question. So basically SSRIs, as I mentioned before, they block the reuptake or pulling serotonin back into the neuron that it left from. And when you see that, you kind of see an increase of serotonin binding to these receptors. And psychedelics actually act as serotonin by binding to these receptors. Actually, if you look at a DMT molecule and a serotonin molecule, they are very, very similar. So if somebody were to take SSRIs, they would almost definitely have some kind of interaction with psychedelics. There hasn't been a lot of research on this. Definitely more needs to be done, but more anecdotally, people sometimes report that they don't have as substantial effects from psychedelics. They either have sometimes more substantial effects, and you could potentially run the risk of creating something called serotonin syndrome, which is when there's too much serotonin or too much of the receptors for serotonin are being bound to, and that can actually be fatal. So there are definitely some things to consider when we're looking at both SSRIs and psychedelics. And SSRIs, again, you have about 15 different types of serotonin receptors. The 5-HT2A is one of those 15 different types. So by blocking serotonin from going back in, the serotonin could bind to any one of those 15 receptors. And you could I kind of think of SSRIs as being like a machine gun that just kind of sprays all over the place. Like you might hit your target, you might not, but you're blowing up the whole wall. Versus I think of psychedelics as more of like a sniper rifle. Like you can pinpoint one direct receptor, the 5-HT2A, target it and activate it. So they have a completely different kind of mechanism than SSRIs do.
The next one is, can you speak again about your background and credentials?
Oh, yeah, sure. So I'm a graduate student at the National University of Natural Medicine. I'm currently pursuing a degree in integrative medicine research. I also have experience working in several different labs at Oregon Health and Science University. I have also done several literature reviews and systematic reviews on psychedelics, and that are coming out right now with more and more research that's coming out. And I'm currently in the process of starting a research project that looks at psilocybin and mystical experiences and its effect on depression and anxiety symptoms. Specifically, I'm interested in looking at what predicts a mystical experience. But we'll see how the coronavirus impacts that study.
All right. Another guy earlier asked about the negative affective bias, and I don't know what the question was exactly, but probably just wanted to have that term clarified.
Yeah, definitely. So negative affective bias kind of plays into the idea that our brain kind of jumps to conclusions about things. Kind of when we have that system regression occur through the developmental period, our networks tend to be kind of clipped out and removed. And this helps us to make quicker decisions about things. Like adults can kind of look at something and almost jump to a conclusion in order to speed our process of this. However, these might not be based on our experiences and things like that, such as racism and other things like that. They may be based upon societal views and other things that kind of mold our perspectives to jump to these conclusions about things.
Okay, great. And then Rhea asks, the person who asked about your background, asked how she can be a participant in your new study.
Just want to thank you for
Oh. So due to the nature of the study, I would absolutely love if I could just give people psilocybin and run the whole study as a clinical trial. Although unfortunately, we're fairly restricted in obtaining psilocybin and we have to maintain legality. So I'm not actually allowed to administer psilocybin to people. However, we kind of thought of a creative way to get around this. It's more so people who are already committed to consuming psilocybin, and basically they will be recruited through trip sitters. So we will have this group of trip sitters and they will go out and basically meet with their clients and then give their clients an opportunity to participate in this study.
I was just going to say, before anyone asks, no, we will not recommend or give you any contact information for said trip sitters. Because they're going to ask. So, no, we won't. I'm sorry.
Yeah, absolutely. Yeah, that's one thing with the IRB. An IRB is called an institutional review board. So whenever somebody does a research project, they have to get it approved by an institutional review board. And the IRB was concerned that if I ran this study, it would incentivize people to consume an illegal substance. So again, I'm unfortunately not allowed to disclose who the trip sitters are, but they will be working with people. And when they work with people, the people will be offered the day of the study or the day of when they're consuming psilocybin before they take it to participate in the study. That way it can be, I have a very good argument that they were already going to consume psilocybin and I'm not incentivizing them to consume it at all. This is just an observational study. And again, unfortunately, as much as I would love to just be like, these are our people, I would love to have more people in the sample, trust me. The more people you have, the more data, fancy analyses you can do and things like that. But until the FDA allows more research on this, unfortunately, I'm kind of restricted in that sense.
Right on. Well, there's plenty more, and I think we can save most of them towards the end. I think one that you could probably handle quickly is there DMT and shrooms, is there any different that we know how they're received by our sensors, our receptors?
Absolutely. So DMT and mushrooms actually are very, very similar chemicals. Actually, psilocybin is not the actual psychoactive chemical in mushrooms. You consume psilocybin, but then through your body's processes, through your digestive tract, it gets converted into psilocin. And psilocin is actually the psychedelic that affects us when we consume mushrooms. And if you look up the chemical name for psilocin, it actually has DMT in it. There's just a few extra methyl groups kind of connected to it and oxygen as well. But the chemicals are very, very similar to each other. It just seems that DMT shares even a closer resemblance to serotonin itself. And because of this, it binds the serotonin receptor very well, and it's extremely, extremely powerful.
Sweet. All right. Well, do you want to...
Sure. Well, I guess we'll get back to it, and we can get to a few more questions after this next section.
It is 8:15, just so you know.
Okay.
And we have no...
Oh, if we could go back a
restriction
few slides.
Oh, that's true. We don't have to be out by any specific.
So, yeah, just, we can aim for whatever.
So now I'd like to discuss the dangers of psychedelics. Often these have been quite seriously purported as being much more serious than they really are pharmaceutically, through kind of these counterculture movements, kind of like anti-counterculture and other things like that. We can see there was one news article that just cracks me up. Girl gives birth to a frog and doctors blame LSD. Pretty interesting stuff. Next slide. So actually looking at the research, when we think of safety for psychedelics, one thing that we like to think about is what's called the therapeutic relevance, or the therapeutic kind of curve. And we could think of that as being the difference between an active dose and a lethal dose. So let's say a substance, you need 10 milligrams of it to feel something, but then 100 milligrams of it will kill you. You want that gap to be bigger because then the substance is safe. If you felt the effects of a substance at 10 milligrams, but 11 milligrams would kill you, that's a very dangerous substance. Versus if you felt effects at 10 milligrams and the lethal dose was 100,000 milligrams, that's a very safe substance. You can consume a lot of it without having the potential to overdose on it. We also look at dependence potential, how addictive a substance is. And we can see on the graph on the screen, it looks at substances' addiction potential and their active dose to the legal dose, which really says a lot about safety. When we think of substances, for example, the active dose to lethal dose for heroin is about 1 to like 5 or 6. It depends a lot on tolerance as well. But then we can kind of see that common substances such as nicotine, alcohol, even caffeine have a higher dependence potential and active and lethal dose compared to LSD and psilocybin. These are actually fairly safe substances to consume when we're thinking of how much of a dose you would need to actually have an overdose on them, and also how addictive they are. Although there's a little bit more to it when you think about safety of substances than just overdosing. There's actually the cognitive effects that happen in psychedelics, which are where the dangers may arise. Next slide. So again, as I was mentioning, there are, even though psychedelics are relatively safe to consume biologically, meaning that you're not going to go crazy, you're not going to overdose unless you take thousands of times an average dose. And I'm speaking specifically to LSD and psilocybin. That is not the case for MDMA and other substances like that. But these substances can have very intense psychological effects, and people have died from either jumping off of buildings, running out into street and traffic, and a whole other things like that. So when somebody consumes psychedelics, we really need to take steps to make sure they're safe, and it's important to be responsible. So if you're consuming psychedelics, I have several recommendations for maintaining that safety. First, do your homework and clean your room. And what I mean by that is you probably shouldn't have a lot going on the day that you're consuming a psychedelic. Maybe kind of take care of your responsibilities. If you have kids to pick up later at 3 p.m. from soccer practice, might not be a good idea to dose right at noon or during the day. And I like to think of a psychedelic experience like a bridge. Once you get on it, you don't really get off, but you rather travel to the other side. And a lot of times people, when they're kind of having these negative or difficult, I'd say challenging or difficult experiences, some people might be like, I want off right now. And they realize they can't, which kind of causes more anxiety. And I kind of think of like telling somebody to just get out of the experience is like telling somebody to jump off a bridge. And we can kind of think of this as like if somebody was at a festival, took a little too much, started acting up, and then police came in, restrained them, threw them in the back of the cop car, I'd kind of quantify that as them jumping off the bridge. Another thing is be safe. Do not drive a vehicle or operate heavy machinery. And it's always a good idea to have a trip sitter present who is preferably not intoxicated or not heavily intoxicated at least. It's always a good idea to be in a familiar area and have a safe space that you can go to if you need to be alone for a period of time. It might be a better idea to try a psilocybin experience kind of in your own home where you're safe, you're comfortable, rather than maybe hiking out into the middle of the woods where you've never been before. And again, I'm speaking from more people who are more naive, or it's kind of their first times doing it. But again, or even high doses, it's always good to kind of have that safe space. And always make sure to test your substance to ensure that it's exactly what you believe it to be. As I mentioned earlier about kind of the research chemicals that are floating around, and there's some real not so great people who are buying them for very cheap and selling them as pharmaceuticals. And there's been a majority of the 2,5-I deaths were from people who bought it and consumed it thinking it was something else. And another thing to consider is difficult is not the same as bad. Somebody might have a difficult experience, but that doesn't necessarily mean it's a bad thing. We can kind of think of somebody, maybe again, going back to the festival example. They're at a festival, they're running around, they're screaming, they're shouting, maybe not hurting anybody, but just, kind of having a difficult time. And that might be really, helpful for them. Maybe they've been told to be quiet their whole life and kind of been holding this stuff in, and it just got kind of let out. So sometimes, as I mentioned before, they're unconscious amplifiers, and they can really kind of pull things out of your unconscious, especially things we need to work on. And if it pulls out something difficult and it's a challenging experience, it doesn't necessarily, it's not a bad thing. Maybe that was something that needed to be addressed and worked on. Next slide. So I also have several recommendations if you are a trip sitter. First, it's always better to practice soft gazing rather than staring them down and direct eye contact. Somebody might kind of feel intimidated by that. Generally in the animal kingdom, like direct eye contact with somebody is almost threatening or intimidating. So maybe kind of like stare at their forehead or kind of like around their head. Also, it's a good idea to kindly remind the person of their surroundings, like, hey, this is great, but let's get off the public road right now. Or, I love your energy now, but let's not disturb our neighbors. Maybe we could reduce our levels by just a little bit. And I recommend it's only good to offer what you can provide. There's been experiences where people from the MAPS Endo project have gone to festivals and somebody was having a difficult time. The person walked up to the person who was having a challenging experience, was like, can I get you anything? And they were like, yeah, I want a unicorn and all these kinds. So it's good to offer what you have. Like, oh, do you want a blanket? Do you want some food, a water? Can I give you a hug? Also, it's good to prevent situations that may lead to future regret for the person. So some common things that have come up are, I want to call my boss and quit, or I want to call my mom right now. A possible response to that might be, wow, that's really great. How about we write down how you're feeling right now? And then if you still feel that way tomorrow, you can do it. And if a person is having trouble on the come down, art is a really useful tool to kind of channel their excessive energy. Again, the MAPS Endo people were saying that they kind of have this tent, they bring people in who are having a difficult experience, and sometimes people are like, I can't sleep. And then they give them some like crayons and an art pad, they start doodling, and they're out like a light in 20 minutes. And I can't stress this enough, you don't have to be a superhero, nor should you be. If the situation is out of your control, obtain help. Like, reach out to the proper authorities to get the person help. Like, I get this is an illegal substance, but it would be much better to kind of have somebody go to the hospital than have a death or any kind of serious injury or any kind of unfortunate thing like that. And when that happens, it's really important that you're the spokesperson. You tell the paramedics like exactly what the person took, how long ago, all these details, and you really kind of act as the spokesperson for him or her. Next slide. So the Zendo project from MAPS kind of has these guides, kind of the safe space idea. It should be a space you're familiar with, you're comfortable with. When trip sitters are working with them, you're not trying to force any kind of experience or anything on them. You're more sitting there and being support. I kind of think of trip sitters as the environmental thermostat. If things are too loud, they kind of tell people, hey, can you quiet down out there? Or if the person needs water, they just make sure the environment is a safe and comfortable place for the person, or obtain help if they need. Through and not down and difficult is not the same as bad. Next slide. So we'll see if this works. This is an audio recording of a case study of a participant who was 17 years old.
If you play it from yours, it should pick up.
Okay. Yeah, the participant was 17 years old, and he consumed what's called 2,5-I, which is a research chemical. And kind of this case really highlights some of the dangers with 2,5-I. Not only is 2,5-I potentially dangerous alone, as it's a very new chemical developed in 2000, or really hit the streets in about 2013, we really don't know interactions with this substance. We don't know how medications that people might commonly take might affect them. So this patient, he woke up in the morning, took his Accutane medication, and then later in the day consumed 2,5-I. And little did he know that there would be a very negative interaction between these substances. So I'm going to go ahead and try and play it. Hopefully this will work out well.
Yeah, so I woke up, I took my Accutane that's prescribed from my doctor, and then about 12 hours later, I decided to try this new drug with my friends called 2,5-I, and I didn't know much about it. I just heard it was like stronger than acid, and I've taken psychedelics regularly. So I thought I could handle it and stuff. I remember when I took it, it made my whole face completely numb, and it just had this really gross metallic taste. It's really like synthetic tasting. But I didn't think much of it. Moments later, I immediately start feeling effects like vibrant colors, jitteriness, like just anxious. And my friend was asking me, "Oh, are you good?" I'm like, "Yeah, I'm all good. I'm just really high, like really fast." And like this was weird because normally psychedelics, in my experience, they've taken like 45 to almost like 90 minutes to kick in, and this was this was almost immediately. So the visuals start getting more and more intense, and I begin to panic. I then start to see like cop lights in my house, and then I get paranoid, like, oh, am I being too loud? Like they must know. So I just tell my friends, like, I'm just gonna crash. I'm just gonna go to bed. I thought sleeping would make me feel better. So I got under the covers, and that's when everything got really crazy. I just kept waking up confused, like each scenario more bizarre than the last. And I remember just not being able to come up with a coherent sentence or thought, and everything was all jumbled up, like a word salad of real and not real words that had no relation towards each other were just forming in my head. And I couldn't communicate to my friends that I was messed up, and I thought I gave myself like permanent brain damage. And that's when I started getting really confused. And yeah, that went on for a long time. And then I heard this really loud static noise that just kept building gradually. It just kept getting louder until it filled the room, and I just felt so much pain and just scared and just terror, and just I thought, this is the end. This is what dying feels like. I'm dying right now. And I felt really guilty and really sad because at the time I was like 17, and I was like a month away from graduating, and I was thinking, oh, how could I go out like this, and how could I let my dad down? And and I think somewhere after that, that's when my friend tried to calm me down, and he tried to offer me a banana, and that's when I got really religious at that point, because I thought I was dying. And at that point I went back to thinking of Adam and Eve, and it's really strange, but when Satan was, trying to get Eve to eat the apple, I kind of thought in the same sense. So I grabbed the banana and I attacked him with it. And then it took about like 3 of my friends to take me down, and that's kind of when I lost myself and went into another void. And and then when I came to, I saw my dad, who now I know that wasn't my dad at the time, but yeah, he was there and he was crying and he was like, my boy, what happened to my boy? And I felt so guilty. That's when I felt just like complete shit. I just felt, it was really, really hard to see your parent cry, especially over you and your dead. And yeah, I saw the whole, I saw the paramedics come in. I got zipped up in a bag, and I saw myself from like a third bird eye view, and it was really real. And I truly thought that I had died, and I just messed everything up, and that was it. And then that I'd just be known as the kid who died at 17 years old. And that went on for just like, it felt like eternity. And it just, yeah. Sorry, I lost my train of thought. Oh yeah. And then I got naked because I thought that's the only way that I could get raptured. That at the time it made sense, but yeah, I got naked and I jumped out of the window, or at least I tried to. I got halfway out, and the other half of me naked just kind of dangled in the living room. And eventually I just came to. What helped was towards the end, I think my sister was playing the guitar, and it was so funny. I was so high and so gone, and no one could get through to me. But my sister playing guitar, singing like a Bob Marley cover, but like she put my name in it. Like it was like Nico, Don't Cry or something like that, in the Bob Marley type deal. But that was the only thing that broke through and made me feel comfortable, which was so bizarre and strange. And I thought I was at my own funeral, my own wake. Yeah, it lasted for at least, they said I was like that for 5-6 hours, screaming at the top of my lungs, and my neighbors laughing, and my sister, defending me. And yeah, okay, that's it. That's all I got. Thanks a lot.
All right.
Oh,
That's fine.
there we go.
Your audio, man.
I have a feeling that those neighbors weren't laughing. So, this patient did a fairly decent job of adding some humor to that, but this was a very serious issue in which there was a very adverse reaction between these two chemicals. Next slide. So 2,5-I is a psychedelic hallucinogen that was developed specifically for mapping the 2,5-I receptor, or the 5-HT2A receptor. There are different, as I mentioned before, psychedelics bind to these receptors, and a drug that binds to a receptor is called an agonist. And there are semi-agonists, partial agonists, and full agonists. And most psychedelics, like even LSD and psilocybin and DMT, are considered partial agonists of the 5-HT2A receptor. However, 2,5-I was developed to be the first full agonist of the 5-HT2A receptor. So it's an incredibly potent chemical, and what they do is they attach what's called a radioligand to this chemical. And they're able to, it glows under radiation. So they basically would give a mouse a high dose of 2,5-I with a radioligand attached, and they can see where it binds to all those receptors, so they can actually map out the 5-HT2A receptor. It was never meant to be used for human consumption. And next slide. There are some significant dangers associated with 2,5-I. It is not the same as LSD, where you can essentially take 100 or 200 hits and not have a fatal overdose. People have died off of as little as 1 or 2 hits. And it seems that as it's a relatively new substance, little interaction is known with other substances, as we've seen in the case with the patient described previously. Next slide. So then there's Accutane, and Accutane is a medication that's used for severe acne, but it also has a black box warning, which is the most serious warning regarding side effects. In fact, Accutane has been known to produce symptoms of depression and even suicidal thoughts. Next slide. So, again, 2,5-I is an incredibly dangerous substance. It actually causes what's called blood platelet aggregation. So blood cells begin to stick to one another, and it causes this blood to kind of slow down, become more sludgy, and can even cause blocks and cause heart failure, which is something you do not see with LSD. And there seems to be a specific interaction between Accutane and 2,5-I, not necessarily between the 5-HT2A receptor, but another one of serotonin's receptors. And it seems they both had an interaction on that same receptor, which kind of caused these incredibly negative effects. Next slide. So this might be a good time for another quick intermission. And then when we come back, we can discuss, I just have some basically ideas and feedback. If you're interested in being a psychedelic researcher or clinician, what are some things that you can do to achieve that?
Okay. I need to use the restroom. Okay. If anyone else says you can use that restroom, and the director else use.
Oh wait, this is the break?
Mm-hmm.
Oh, break.
I can answer questions, though, if people have them.
Let me see what is going
I
on.
wrote down a bunch, but
In code.
that paper have it?
Take a look. What the heck, Jason? Yeah, I see it. Give me 1 second. Okay. So,
What are the significant differences by microdosing LSD and mushrooms was one.
No, okay, so yeah, one of the questions was what are the significant differences between microdosing LSD and mushrooms? Pharmacologically, again, there's not been a lot of research on comparing different psychedelics to one another in that regard. Actually, one of my colleagues is proposing a study to do that, so hopefully we'll see more of that coming out soon. But if we're talking about the actual substances, I believe LSD is a little bit easier to microdose with, and that's because you kind of have like this set amount on a blotter paper. You can kind of cut it into different areas, versus mushrooms kind of can have variations. I mean, obviously between strains, but even the same strain, depending on where it's grown, how it's grown. If somebody's picking wild mushrooms, they might pick some from one area, some from another area. They may have different potencies, so it can be a lot harder to microdose with pure psilocybin, with mushrooms instead of pure psilocybin. But people definitely find a way. It's 100% possible. It's just kind of what works for you. I would believe that it would be much easier to kind of reach that threshold period of not a microdose anymore to actually experiencing effects if you were to consume mushrooms, just because the way that the substance is presented.
got it. And another question. Yeah, you got other ones?
Yeah. Why are psychedelics kept illegal if they have no harmful effects?
That's a excellent question, and I wouldn't go so far as to say they have no harmful effects. For example, in the case study we just saw, even though that was a research chemical, definitely that people have had experiences like that on LSD, and these experiences can be traumatic. In fact, that particular case had a little bit of PTSD following his experience. So they definitely do carry risks with them. So I wouldn't say they're completely harmless, but they're definitely much less harmful than a lot of common substances we have out there, like alcohol and nicotine. And I mean, I don't want to go on a rant too much about that, but that really has to do with the Controlled Substance Act. It's clear that substances are not made illegal or illegal based on any kind of biological reasoning. They're based on societal reasoning. It's what society thinks is appropriate, what society thinks is okay. And for example, alcohol has the worst withdrawals of any drug, even worse than heroin. Somebody could essentially die if they were a heavy drinker and they cut using alcohol. Nicotine is the most addictive substance. It's even more addictive than crystal methamphetamine. When we mean by addictive is people who use nicotine have a much harder time quitting and generally use it for a majority of their lives. Both alcohol and nicotine, which are completely legal, are kind of exempt from the Controlled Substance Act scheduling and things like that. And it seems that a lot of substances are made illegal to kind of target different groups, to kind of like go after them almost. A way to make, if the government is not happy with a certain group or population of people, a way to make them all criminals overnight is to illegalize the substances that they're using. We saw this when marijuana first came on, and there was lots of immigrants from other countries coming in who consumed it. By making marijuana illegal overnight, all of the immigrants were criminals who used it. Also, there's political motives around that, and economic ones as well, like from the timber industry. And then there was, in the 60s and 70s, there was the whole countercultural movement. Vietnam was going on. A lot of people were very unhappy with the war, and a lot of these people were consuming psychedelics. And the administration felt that a way to kind of get rid of that problem would be to illegalize all of those things. So again, it really seems that it's societal based how drugs are made illegal or legal, and it's not based on biology.
Okay, I got a couple more. There's still more rolling in too, but somebody asked, is serotonin syndrome possible with psychedelics since they vastly differ in their intracellular effect than serotonin?
So again, it is possible, but it's much low likelihood. As I was mentioning before, SSRIs kind of act like a machine gun. They just dump a bunch of serotonin into the cleft, or not really dump it, but prevent it from getting sucked back in, versus psychedelics seem to target primarily one, but a few other receptors. There is a risk of serotonin syndrome, but very unlikely if somebody's consuming a classical psychedelic, especially within what's considered a standard recreational dose, even if it's strong. However, taking medicines like MAOI or SSRIs, MAOI inhibitors, all kind of things like that can actually increase a person's risk of having serotonin syndrome from consuming psychedelics.
Great. Looks like some of the stuff is being answered in the chat, but here's one. Any significant differing effects of psychedelics on a fully developed brain versus a younger person?
That's, again, I wish we had the research to those answers. Those are questions that we really need to kind of be developed. And that's an interesting thing that's kind of been brought up, is like the whole idea of psychedelic assisted therapy. When is an appropriate time to allow people to partake in it? Is it 18? Is it 21? Is it younger than that? I've seen a lot of different researchers with different perspectives. I remember one of the researchers was saying that, it would be okay for people under the age of 18 to consume psychedelics and be part of psychedelic therapy, and even consume them with their family members as a bonding experience. And other people kind of caution against that a little bit more. Like, there's kind of differing perspectives on how that works. And when the brain is going through the adolescent phase of development, there's just so many changes that are going on. Again, we don't know if there's any detrimental effects, or if it improves things, if it makes things worse. We kind of really don't have that knowledge. Even for not psychedelics, but almost all substances. In fact, in one of the labs I work in, that's our primary thing, is we're looking at adolescent brain development, and we look at how different drugs, mostly alcohol, nicotine, and marijuana, affect adolescent brain development. And the principal investigator, the researcher who's in charge of all that, says, we really don't know. Yes, research has shown that, okay, probably consuming alcohol is bad for adolescent brains. We see all these changes that occur. But I guess a better way of phrasing what I'm trying to get at is to really conduct this research you would need 2 alternate dimensions with the same person in each dimension, and then one of those versions of the person consumes the drug, the other does not. And that's the only real way to know with 100% certainty what these factors are, because there's so many other factors that come into play. Like maybe kids who are in higher access communities have other stresses that are also affecting their brain negatively, and all of these other factors to it. So it's really hard to say. And again, it's impossible for research to do that because we don't have 2 alternate dimensions. A lot of research looks at siblings, like, okay, one sibling consumes this drug, the other does not. They're about the same age. It's kind of the best thing we have, but again, siblings can differ quite dramatically.
Okay, one more. This guy says he has a set schedule and his circadian rhythm is out of whack due to his, he's coming off methadone. And he asks, is that a serotonin shortage, and should I microdose?
So the circadian rhythm definitely does have a major role in serotonin. Actually, serotonin is converted by the pineal gland into melatonin, which I'm sure quite a few people are familiar with. Melatonin is a substance that helps people go to sleep. But personally, whenever I take melatonin, sometimes it works, but it's not that effective all the times compared to some other things. And it turns out there's the blood-brain barrier, and melatonin has to fight against all these other amino acids that are trying to enter the blood-brain barrier. So when it's actually produced in the brain, though, it's incredibly potent and incredibly effective. And like, for example, at night, they always tell you shouldn't look at computer screens for a while before going to bed. That's because when it's dark, your brain starts converting serotonin into melatonin, and that helps you to sleep. And if you were to walk into the bathroom in the middle of the night, flip on a light, all of a sudden that cancels that process, your melatonin stops, and you start waking up. So it's hard to say exactly because necessarily you'd see an increase in serotonin, because if there'd be an increase in serotonin, then it can be converted into melatonin. Psychedelics wouldn't necessarily increase serotonin. They pretend to be serotonin. They act like serotonin and bind to the receptor. So I would probably recommend, rather than psychedelics may be helpful, but maybe something that increases serotonin. I might recommend like a combination of maybe like melatonin or 5-HTP, kind of other things like that are just increased serotonin. Another interesting thing, just while I'm on the topic, is there's tryptophan, which is commonly found in turkey, and that's actually a precursor of serotonin. So that's generally why when people eat a lot of turkey, they take in the tryptophan, which converts into serotonin, and then into melatonin, which then makes you sleepy.
Sweet. Okay. Yeah. Well, do you want to get back in it? The last
Sure.
question that was remaining, I think, is covered here, and that's what academic paths can one take?
Oh, that's what we're about to cover.
Okay. Right on. So I thought that would be a good one to hang on to. Okay, let's see here. What happened here, Evan? Did this close, or?
Oh, yeah, I pressed the wrong button. I'm really sorry.
That's all good. I was just like, where the heck did it go?
I am not the master of the computer.
Okay.
One sec, let me just make sure it's on. Whoa. Okay, and then we'll move you over here. And let me just make sure that this is responsive here. Yes. Okay, we're back on.
Okay. So now I'll go over a few pieces of advice that I could give to anybody who's interested in being either a psychedelic researcher or clinician. I am biased towards research, so I do have a lot more advice for researchers than clinicians, but I do have advice for both. Next slide. So psychedelics are still heavily stigmatized. As of yet, there is no obvious infrastructure for which enthusiasts can channel their energy. Not the response you wanted to hear, I know. We're getting to it, though. There are currently no psychedelic research graduate programs, although there is a certificate program, which I will talk about a little later. There's no psychedelic student groups, there's no psychedelic scholarships, and few professors are willing to provide mentorship or funding. and not many people are willing to sponsor psychedelic research currently. And on top of that, you cannot currently get federal funding. All the current psychedelic trials that are going on are being funded by donations. Next slide. So first, it's important to think about why you're interested in being a psychedelic researcher. Is it because you think psychedelics are novel and cool? Well, you're probably going to be pretty bored because this is moving very, very slowly. And it's very kind of tedious work that sometimes is not very rewarding. Is it because you had a mystical or life-changing experience on a psychedelic? Well, you don't need to be a researcher to continue having those. Is it because you're frustrated living in a culture that tramples individual freedoms, discourages introspection and insight, and that you want to instead change the society for a better? Well, there are probably different ways and more effective ways to kind of be an advocate for change and for better things than to be a researcher. As we can obviously see from like the Controlled Substance Act, the government does not always listen to research. Next slide. Or are you interested because you are motivated by a genuine curiosity about these particular substances and wish to apply the modern tools of inquiry towards understanding their properties? Maybe you know that the discovery of LSD is what sparked interest in the serotonin syndrome and prompted the explosive growth of modern psychopharmacology that continues today. Maybe you contemplate what other wonders may lie hidden in the closed box of psychedelic science. I think that is probably one of the best reasons to be a psychedelic researcher. Next slide. So there are some things to consider if you're still willing to pursue this path. Are you willing to accept that your unconventional interests may lead to professional isolation or even ostracism? And that time-consuming navigation of the layers of the red tape endemic to psychedelic research will inevitably slow your publication rate and promotions compared to your peers? Are you aware that the total lack of government and corporate support for such endeavors means you will never be rich, and you may even one day end up in jail on charges of one sort or another? Next slide. If you're okay with that, then by all means. As an undergraduate, get your degree and lie low and infiltrate the system. Educate yourselves about psychedelics, such as what you're doing right now. Start a psychedelic student group if you're part of a school or an organization. Volunteer. We could definitely use some volunteers here with the Portland Psilocybin Society, or Psychedelic Society, and a whole bunch of other amazing places to volunteer. You could write letters to congressmen or people in power. And as a graduate student, a great first stop would be the Hefner Research Institute Scientific Advisory Panel, which is a list of psychedelic allies in the international academic world. Next slide. So this is a more direct answer to the question that was proposed earlier. So if you wish to follow the PhD route, pure neuroscience or neuropharmacology is extremely valuable. Experimental psychology, or the study of the human mind, is also very valuable. Clinical psychology is also very valuable, and it will allow you to build the skills necessary in a multidisciplinary team that is researching the psychotherapeutic value of psychedelics. Also, it'd be interesting to consider psychoanalytic training, which is not just for MDs anymore. So learning to navigate the subconscious mind can be a very valuable skill for anyone who wishes to do psychedelic psychotherapy. Even cognitive science, a pure science of the mind, drawing from a variety of disciplines, including computer science, is very valuable. However, this is not a complete list. There are still many, many other degree paths that would be useful in psychedelic research. More advanced studies are starting to use machine learning, so any computer science degrees would be beneficial. Even more like English degrees, science communication degrees. There's many different ways that one could kind of be valuable and kind of support this research. Next slide. So, in the end, there are many paths to becoming a psychedelic researcher, but what matters most in the end is that you obtain success and satisfaction on a personal, professional, and spiritual level, while at the same time remaining true to yourself. Next slide. So, unfortunately, I only have one slide for anybody who wants to be a clinician. As I mentioned before, I am a little biased. So if you're interested in being a clinician, I recommend exploring training programs and degrees in transpersonal oriented psychotherapy, which seems to be the common model used in most psychedelic assisted therapy models. Currently, the California Institute of Integral Studies offers a certificate in psychedelic assisted therapies. I believe the program is about $10,000 off the top of my head. However, most organizations such as MAPS and a lot of the places that are doing research requires clinicians or researchers to go through this training program.
Compass, Pathways, and USONA Institute have their own training, but it's not open to the public yet.
Got it. Yeah, and as Evan mentioned, Compass also has training programs, but not all are open to the public. This particular one is. Although you would have to already have a degree, I believe, somehow related to psychology.
Are you talking about the, sorry, I blanked out for a second, CIS?
Yes.
Okay, you do not have to have a degree related to psychology.
Oh, but okay.
But you do have to have
A degree, yeah. So there's also MAPS offers an MDMA training program as well. And even more so, like there are more and more research institutions that are popping up. And even if you're not interested in being a researcher, clinicians that can kind of go monitor and provide the therapy are very, very useful in the research setting. For example, Johns Hopkins just established the largest research psychedelic center in the world. They got a $16 million grant to do so, and the Imperial College London as well has a research institute. So kind of exploring pathways like that may also be a good idea. Furthermore, the MAPS has a Zendo harm reduction unit called the Zendo Project, which offers peer support volunteer training. And once you kind of gain that experience, you can go to festivals, maybe not now with the whole virus issue, but when that clears up, you can go to festivals and kind of help people who are going through difficult experiences, which is an amazing thing to kind of have on your resume if you're interested in doing psychedelic clinical work. You can also start your own practice as a holistic therapist. There's kind of this common theme where therapists, like with Ibogaine, tend to set up clinics in Mexico or other countries, and then potentially participants will fly down there. There are some significant barriers to that, though, like not everybody can afford to do those things and other things like that. So, and there's another, essentially, if you're interested in being a clinician or you are a clinician and you want to start doing this work, you can start talking to people, not prescribe. I do not recommend that any clinician prescribe psychedelic therapy to anybody currently, because you could lose their license. In fact, in Denver, Colorado, where psilocybin was decriminalized, there were a few therapists who were doing psychedelic assisted therapy even after it was decriminalized, and they lost their license. So I do not recommend prescribing them to people, but if a client wants to come and talk to you about their experience, there's absolutely nothing illegal about that. And above all, I highly recommend practicing self-care and stay within the law to avoid unnecessary consequences. And at a last meeting, there was a person who brought up a very good point. While I recommended that we stay within the law, they disagreed with me, and they said that people are dying every day from depression, addiction, PTSD, and we have the tools to help them now. And we can't wait for the government to get its head straight and legalize these substances, and we need to do it underground. I'm not going to ever recommend that anybody does anything illegal. There are pros and cons to both. We could save lives, but one slip up and this whole renaissance could go under. So there are things to consider, and just really do what you believe is true to you.
I had a few things.
Yeah.
You have some like.
I guess it is sensitive.
I think that's the last slide.
Yeah. So, and I just wanted to say a few things as well, because I personally am very interested in becoming a psychedelic therapist or clinician. Anything by Stanislav Grof I highly recommend. He's an extremely well-versed therapist. He did a lot of the research in the late 50s and early 60s and has been working with psychedelics and other modalities of consciousness ever since. He's still alive and teaches at the California Institute of Integral Medicine, or California Institute of Integral Studies. That's what it is. Thank you. But yeah, any of his work is a great resource. I also recommend Leo Zeff. The Secret Chief Revealed is a book about sitting with people. I shared just now in the live chat a article from Psychedelic Times titled, I forget what the exact title is, I got it right here, How to Become a Psychedelic Therapist, From Clinical Trials to Integration Therapy and Everything in Between. And it's a great resource with pretty much all the information you could ever want for. I will also say that particular areas of study in psychology, things like transpersonal psychology, humanistic psychology, holistic therapy is a big, thing in the psychedelic integration industry, and I said transpersonal, right?
Yes.
Yeah, okay. Transpersonal, humanistic, and psychodynamic, existential. Also, it's extremely important to be well versed as a psychologist on trauma and what trauma does to people and how to relieve it. That's all I got, pretty much.
Oh, speaking of trauma, that's
Sure.
another important.
I'm gonna pass it back to Aaron.
So yeah, absolutely. Trauma is a very important one where we have to consider in these studies. And there's been some reviews that have come out that have looked at racial inequalities in psychedelic research. A review that looked at 17 different studies from 2000 to 2017 found that I believe it was 89% of participants were Caucasian, and only 2.2% were African American, 2.2% were Latino, and 1.5% were Asian. So there's a very, very low level of inclusion for people non-Caucasian. So, and that's a major issue. That's something we really need to address, because people, are different, especially if we're talking about with trauma. We don't really understand how historical and racial trauma and all these things impact psychopathologies, and how psychedelics, how they all relate to one another and their interactions between each other. And there's really a lack of understanding of how trauma, especially racial trauma and things like that, really impact psychedelic research. So we really need to do more studies on that. And it's hard to. I'm not a person who points fingers or blames people. I'm not necessarily saying it's the researchers' fault or anybody else's fault, but there's definitely been kind of unfair drug policies in the United States, especially around substances. Generally Caucasian people, in the 60s, would go around advertising their psychedelic use with much less consequences than people who are not Caucasians. And rates of incarceration and all of these things are very, very substantial. So it might be that non-Caucasian people feel scared to participate in a psychedelic studies, where it's kind of this illegal substance that they've been treated much more unfairly around than the other people who are doing it. There was this article that I saw a few weeks ago that had these Caucasian women doing yoga, and the article was cannabis really helps out with yoga, and that's what the whole article was about. And people were commenting wow, so Caucasians get to oh, this is a fun thing to do, but then non-Caucasians go to jail for it. So that's serious things that we need to consider when we're looking at this research.
Right, okay, back to QA.
Yep, so.
Got a few minutes, we're after time, so, we're at your schedule now. If you just want to go home, we can
I can answer
a few more.
Yeah, I can answer a few more questions.
Right on. Okay, we got a few more for him then. Could you please cite your source for that graph you showed with the drugs and their addictive potential?
Oh, absolutely. If you take my email down, send me a message. I'd be happy to send you that source.
Okay, great. Here's the other one. Salvia operates on the K-opioid receptor.
Yes.
Does that make it a true psychedelic? And how does the K-opioid receptor even give you a psychedelic effect?
So it doesn't just affect the K-opioid receptor, but that was kind of an interesting thing. I recently went to one of the largest neuroscience conventions in the world, and they were talking about how they found salvia acts on the opioid receptors, which was a very odd thing. And it's not generalized as salvia only affects the opioid receptors, which is why it has these effects. Salvia affects a whole bunch of other different receptors as well. Kind of one thing that maybe I should have explained more clearly, all psychedelics, not just the classicals, impact the 5-HT2A. So salvia not only impacts opioids, it's the 5-HT2A, the NMDA receptors, as well as a whole host of other receptors. So it's this perfect cocktail of different receptors that kind of has those effects. But it's a pretty good idea, or it's pretty logical that the opioid receptor, which is responsible for kind of pain, pleasure, breathing, other things like that, gets kind of interrupted on more of a dissociative where somebody doesn't feel connected with their body. There's kind of this numbing effect, which kind of promotes the whole like being pulled out of the body effect when it deals with the parietal lobe as well.
Okay, right on. Here we go. Last one, I guess. Unless anybody sees this now and types in your question. In clinical trials, what have the adverse reactions been to any psychedelics?
They've generally been fairly mild. For example, in the Robot and Carhart-Harris study where they had the 20 participants, they monitored adverse reactions. Every single participant experienced mild transient anxiety before consuming the psychedelic, and some during and some a little after. Another side effect was headaches. So, and again, these were the scale rated not only what the adverse reaction was, but how long it lasted and how intense it was. So all of the adverse reactions were short and not intense, but they were anxiety, headaches, and paranoia were the three common ones. No participant needed any kind of tranquilizing medicines, which they had on staff in case something negative happened. They would go and administer those tranquilizers. No participants required that, even on the high dose.
Righteous. You got anything more, Evan?
I'm good to go.
Okay,
Yeah.
I got just one final one, kind of towards the end of your thing. You said no one's funding psychedelic research, but I read there was like a multi-million-dollar institute built somewhere on the East Coast. Was it Johns Hopkins?
Yeah, there was. So what I mean,
In the bathroom when you talked about it.
generally research is funded. Most all research is funded by the federal government through the National Institutes of Health. There are different types of grants. There's an R01, which is a research grant. There's K grants. There's T grants. There's a whole variety of different grants. And what I meant by that is the National Institutes of Health are currently not providing funding for psychedelic research. So like the Johns Hopkins school, MAPS, like everything like that has been through donations. For example, Bronner's Soap, if I believe it was them, donated $5 million to MAPS to carry out their trials. So it's not necessarily the government that's paying it, but it's just people who very much believe in this work and are willing to put down millions of dollars to help support it.
Okay. And maybe this one will just be the good one to end on unless something else comes through. How can we, as just normal people, best support psychedelic research and contribute to the field?
Absolutely. That's an amazing question. Well, probably the first one is to, whenever you consume a psychedelic, make sure you're safe. And take, as I mentioned before, have those steps in place, because the more people who are ending up in emergency rooms or hospitals or injured or dying around, like not from the actual psychedelics interaction, but from like doing things under the influence of psychedelic, that'll set the whole field back drastically. So really just making sure that we show people we're responsible, people aren't ending up in hospitals from these substances, and that, prove them wrong with actions, kind of a thing. And another thing that could be helpful, if you ever see an opportunity to enroll in a study, that would be greatly appreciated. We need more participants, other things like that. And I would say probably the last and maybe even the most important thing that you could do to support psychedelic research is just talking to people. Like just sharing what you learned here today with a friend or a coworker, or just kind of like spreading the word, and like really basing it around the facts. Like we don't need to spread rumors or anything, but just be like, hey, there was this study that showed blah, 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 and just kind of really, maybe keeping up to date with the research that's coming out. I know kind of a major negative about a lot of research is the journal articles are generally behind paywalls, which is really unethical in my opinion, because we pay federal money, not for psychedelic research, but for most research. And then the journals take them and then they make you pay $60 to read the article. So you're paying twice for it, which is highway robbery. And the idea behind that is the journal proofreads it to make sure everything's okay. But, they're behind all these paywalls, and those news articles you see don't always translate it that well. So there are some kind of issues in that, but exactly what you're doing right now, kind of like reaching out and going to Portland Psychedelic Society events and other things like that, like the upcoming conference, just learn about them, keep yourself updated about them, and just share and talk and spread the good word with people.
Yeah. Okay. Could I add to that?
Please.
Yeah. So I agree very much with everything that Aaron just said. I just want to reiterate the importance of getting out and talking about these things, helping, the Psychedelic Society and all of its members destigmatize the use of psychedelics. Psychedelic Society exists because people don't feel comfortable going to work after a weekend where you had a mushroom trip, and when someone asks how your weekend was, you don't feel comfortable saying, I had an amazing, mystical, life-changing experience on mushrooms, and I lost my sense of ego, and I traveled to a different dimension, and I healed myself, and I came back, and I'm okay. How was your weekend, Janet? It's like that's not really a acceptable thing, but do it anyways. If you do it, it'll start to become more acceptable, and there's no denying the results of the research that are coming out every other month all over the world. So there's really no reason that you should feel ashamed or guilty about healing yourself with these medicines, the same way someone shouldn't about using a cup of coffee to get them through a work day, or a glass of beer to help wind them down from a work day. We need your individual help to spread the message. And like I said before, the Portland Psychedelic Society was started by a single woman that was inspired by her experience with psychedelics. Shout out to Helen. And you have the power to create this container. I will say I recommend that you practice self-care and make sure that you can take on the responsibility of holding space for other people, because it's not as easy as it sounds or looks, but it's not really that hard, Like I said, the Psychedelic Society does not have to be as big as the Portland Psychedelic Society. If you're in Little Rock, Arkansas, and you want to start one there, schedule a meeting at you can't now, dang it. But, when the coronavirus passes, or schedule some online meetings. And, be careful, but have hope.
Right on here, I'll do it.
Okay.
I'll sign us off unless anybody has anything else.
Did you want to say something else?
Yeah, here we go. I'll take it back there.
Just one comment to that. Like, even if it's not research that you're telling them about, it can be anecdotal. Like, talk about your own experiences, like what Evan was saying. Like, if you had a really amazing experience or even a challenging experience, talk to people about it. Kind of like let them know what the reality of it is, because definitely a lot of people either who have never taken them before They just don't really understand what psychedelics are, and the media is no help with that. They really kind of purport everything around that, the Controlled Substance Act, legality. So it's really just like breaking away the stigma, like kind of all how psychedelics are embedded in our society, and really like presenting them as what they really are, these medicines that have been used long before our society or any society was a thing.
All right. Thank you, Aaron. Thank you, Evan. I'm going to turn the camera onto me now and just close this up. Okay, that's good. Yeah, let's see if we can just get it to stop spinning. Okay. All right, yeah, I'm just going to add to that. You know, it is COVID lockdown for most of us here, and so we can't start our own psychedelic societies, but this gives us a great opportunity for everybody worldwide to come in and sit in integration circles with all of the communities that are doing it. And you can do some tremendous networking right now, not just meeting Portland people, but Baltimore, Dallas, Minneapolis. They're all talking about doing joint events now. And who knows where this is going to go. I mean, this is really going to open it up for small town folks and worldwide folks to come in and sit in on these meetings and participate. So I wanted to say that, you know, ours have not been canceled. We're not longer meeting in, you know, in real life, but everything is still going to carry forward on schedule. We're going to do our support groups on Zoom. We're going to do our presentations here on live stream. And, you know, we're just going to keep the information going out. You can support this channel right now. The only way is by PayPal. Or you can email me at that ppslivestreams@gmail.com, and maybe we can try to figure out another way. Also, we have these cool t-shirts. They're not exactly for sale online, but if you really, really want one, hit us up at that email. They will usually for sale at our live events, but since we don't have any, we have 200 of these t-shirts, and I don't know, $25 plus shipping. Stickers are by you. Oh, there's one on my, there. Yeah.
Look at that.
Stickers, write us, make a donation if you have a PayPal. That would be really cool. It would mean a lot that we, you know, touched you tonight and that we got some feedback. Also, all feedback, collaborations, all that stuff, just send it to that email, ppslivestreams@gmail.com. And look forward to trying to put something else online this week. Do we have anything coming up real soon?
The mushroom video is going to be out within the next week or two.
That'll be over here at my house?
Mm-hmm.
Okay.
Yeah. Information on how to start your own psychedelic society is hopefully going to be coming out in the next month. But you're not going to be able to do much with that for a little while. So.
Okay.
Like we said.
Well, we're going to be learning, you know, follow us on social and stuff, because we're going to have another learning curve here with Zoom. We're talking about opening up a Zoom meeting that's going to be 24 hours, where it's just going to be a lounge. Come on in, say hello, make sure your equipment's working, ask any pertinent questions, and then hang out. Else, you know, no one's going to be there for 24 hours. I'm going to start it up. I'm going to be here for like a couple hours, and then I'm going to go over to Joe's house and get his Zoom set up at his house,
Nice.
Join him in, you know, and then hopefully there's just people. What the idea is that a lot of people haven't used Zoom before, and you have a little just technical hiccups the first time you have to do it. So we'll give you a 24-hour window where I'm going to be providing tech support, letting you get familiar with the damn software, really bringing you in, because I'm doing event organizing this year for PPS, and so we canceled all these events, but we had 180 events planned that we are not going to be able to do in person. So that's going to give us a tremendous outlet here online. So, all right. Thanks for sticking with us. Thank you, Aaron, for a wonderful presentation. We're all social distancing. Evan. All right, guys. Well, thank you very much. We'll be back with something more for you here in the next week or so. Evan's going to do a live mushroom demonstration for online only.
Bye.
Peace.
Bye.